Good day, everyone. We are the undergraduate plant biology students of the Mindanao State University Iligan Institute of Technology of the College of Science and Mathematics under the Department of Biological Sciences. I am Mark Joseph G. Portusilio, and together with my colleague, Riami Dalumbar, we will be discussing to you the topic entitled Enzymes in relation to the Plant Physiology and Development Botany 101 Report. In this report, we will be discussing the following key points to keep us on track and organized. Kindly have your papers prepared to take note of the important lessons that you need to remember and familiarize. Today, we will be knowing the biological molecules, typically proteins, that plays a vital role in our lives, that serve a wide range of important functions in the body. These are what we call enzymes. Enzymes are defined as catalytic proteins that increase the rate of biological reactions within the cells of our body. Enzymes digest our foods, contract our muscles, and synthesize hemoglobin. They catalyze reactions that supply all necessary biomolecules and energy for our survival. Enzyme can also be defined as catalyst that increases the reaction rate by changing the way a reaction takes place, without itself undergoing any net change. Let's take into account that an uncatalyzed reaction in a cell may take place eventually, but not at a rate fast enough for our survival. For example, the hydrolysis of proteins in our diet would eventually occur without a catalyst, but not fast enough to meet the body's requirements for amino acids. The chemical reactions in our cells must occur at incredibly fast rates under the mild conditions of pH 7.4 and a body temperature of 37 degrees Celsius, which means that enzymes are biological catalysts responsible for supporting almost all of the chemical reactions that maintains our homeostasis. As catalysts, enzymes lower the activation energy for a chemical reaction. Shown here in Figure 1, the enzyme carbonic anhydrase lowers the activation energy needed for the reaction of carbon dioxide and water. Less energy is required to convert reactant molecules to products, which increases the rate of a biochemical reaction compared to the rate of the uncatalyzed reaction. For example, carbonic anhydrase, an enzyme in the blood, converts large amounts of carbon dioxide and water to carbonic acid. In the presence of the enzyme, this reaction occurs 10 million times as rapidly as it does in the absence of the enzyme. One molecule of carbonic anhydrase can hydrate about 1 million molecules of carbon dioxide a second. For a better analogy, let's put you in a situation where you're about to jog. If you'll jog in kilometers wearing only an uncomfortable pair of slippers, you'll be going slow and it'll be hard for you to reach your destination at your desired time. So let's symbolize your pair of shoes as your enzymes, which makes you jog more comfortably, faster, and can help you reach your destination with your desired time. There are two types of enzymes. The first one is called simple proteins, which consists only of a polypeptide chain. It is the tertiary protein structure of the simple enzyme that makes it biologically active. Second is called conjugated proteins. Its protein portion is inactive without a cofactor. The cofactor is the non-protein portion of an enzyme, such as a metal ion that is necessary for enzyme activity. If the cofactor is an organic compound, usually a vitamin, it is called a coenzyme. Examples of conjugated proteins are apoenzyme, the protein portion of the enzyme which is inactive. Next is the holoenzyme, a combination of apoenzyme and cofactor, which makes it an active enzyme. Lastly, the cofactors. Cofactors are organic compounds derived from B vitamins or called coenzymes and inorganic metal ions. We will be knowing further about these enzymes as we go along with our report. Here in Figure 3a, the apoenzyme is unable to bind to its substrate. The Figure 3b, the required cofactor in this case a copper ion is available. It binds to the Apple enzyme. Now the active site takes on the correct configuration. The enzyme substrate complex forms and the reaction occurs. Next up, we'll be learning about the enzyme nomenclature. Enzymes are named according to the type of reaction they catalyze in their substrate, or the type of reaction they catalyze or their substrate. We all knew that substrate is the reactant upon which the specific enzyme acts. Enzyme physically binds to the substrate. Enzyme are commonly placed with a suffix of ase or ase, such as lactase, amylase, lipase, or protease. This denotes an enzyme. While some enzymes, digestive enzymes, have the suffix in or in. These are pepsin, trypsin, and chymotrypsin. These enzymes were the first ones to be studied. The prefix denotes the type of reaction the enzyme catalyzes. For example, oxidase. This means it's a redox reaction. Next example is hydrolase, which means an addition of water to break one component into two parts. The substrate identity is often used together with the reaction type, such as pyruvate carboxylase, lactate dehydrogenase. Your substrate for this is the pyruvate and lactate. There are classes of enzymes. More recently, a systematic method of classifying and naming enzymes has been established. The name and class of each indicates the type of reaction it catalyzes, and there are 6 main classes of enzymes as described in the following slides. The first class of enzyme are called oxidoreductases. Its general reaction is for oxidation or reduction. Example of oxidoreductases is called oxidases, which the reaction catalyzed is for oxidation. Next example is dehydrogenases, which the reaction catalyzed is for the removal of hydrogen. And lastly, the reductases, in which the reaction catalyzed is for the addition of hydrogen. Examples in metabolism are dehydrogenases catalyze reactions in which a substrate is oxidized or reduced. The next class of enzymes are called transferases, from its name, transfer. Its general reaction is to move a functional group. Remember, a functional group is a collection of atoms within molecules which bind together to react in predictable ways. Examples of functional groups include the group of hydroxyl, ketone, amine, and ether. Example of transferases are transaminases, which catalyze the transfer of amino group. Second example are the kinases, which catalyze the transfer of phosphate groups. The next class of enzymes are hydrolases. Its general reaction are the hydrolysis of bonds. Examples of hydrolases are peptidases, which reactions catalyze is to hydrolyze peptide bonds. Next up are lipases, which the reaction catalyze is to hydrolyze ester bonds in triglycerides. The next example is amylase, in which the reaction catalyze is to split 1,4 glycosidic bonds in amylose. And the last example is phosphatase, in which the reaction catalyze is to hydrolyze phosphate groups. The next class of enzymes are called lyases. Its general reaction is to remove small groups to form double bond or to add small groups to double bonds. Examples of lyases are the carboxylases, in which the reaction catalyzed is the removal of carbon dioxide. The next example are dehydrases. The reaction catalyzed is the removal of water or H2O. The last example would be deaminases, which the reaction catalyzed is the removal of NH3. Examples in metabolism for lyases are the decarboxylases, in which they catalyze the removal of carboxyl groups. The next class of enzymes are called isomerases, in which we are all familiar with. Its general reaction is to rearrange the atoms of a substrate. Examples of these enzymes are isomerases, in which the reaction catalyzed is the conversion of cis to trans, such as ketos to aldose. The next example would be epimerases, in which the reaction catalyzed is to convert D to L isomer. Examples in metabolism are isomerases, in which they catalyze the conversion of an aldose to ketose. And next would be mutases, in which they transfer the functional group from one atom to another within a substrate. Next class of enzyme is called ligase, in which the general reaction is the synthesis of larger molecules. Examples of ligase are what we call synthetases, in which the reaction catalyzed is the combination of two molecules. The next example of ligases are what we call carboxylases, in which the reaction catalyzed is the addition of carbon dioxide to a substrate. The example in metabolism are the synthetases, in which they link two smaller molecules to form a larger one. Enzymes do also have characteristics. We all know that enzymes do nothing but speed up the rates at which the equilibrium positions of reversible reactions are attained. In terms of thermodynamics, enzymes reduce the activation energies of reactions, enabling them to occur much more readily at low temperatures, which are essential for biological systems. The basic characteristics of enzymes includes almost all enzymes are globular proteins, consisting either of a single polypeptide or of two or more polypeptides held together in quaternary structure by non-covalent bonds. Second, almost all the enzymes are proteins, and they follow the physical and chemical reactions of proteins. Enzymes are sensitive and labile to heat. Fourth, enzymes are water soluble. Fifth, enzymes could be precipitated by protein precipitating agents such as ammonium sulfate and trichloroacetic acid. Enzymes do also have certain properties. According to Cooper, established last 2000, like all other catalysts, enzymes are characterized by two fundamental properties. First, they increase the rate of chemical reactions without themselves being consumed or permanently altered by the reaction. Second, they increase reaction rates without altering the chemical equilibrium between the reactants and products. In accordance also to Infinita Biotech information, there are four most important properties of the enzymes. These are the following: the catalytic property, the specificity, the reversibility, and the sensitiveness to heat, temperature, and pH. The catalytic property of the enzymes, which they have extraordinary catalytic power. They are active in very small quantities. A small amount of enzyme is enough to convert a large quantity of substrate. The enzymes remain unchanged after the reaction. The turnover number of enzymes ranges from zero point 0.5 to 6 million. Turnover number is the number of substrate molecules converted by 1 molecule of enzymes per second when its active site is saturated with substrate. The next property would be specificity. Enzymes are very specific in their action. Particular enzymes act on particular substrate only. Enzymes are also specific to a particular type of reaction. In some rare cases, the specificity may not be too strong. Enzymes show different types of specificity, which will be discussed later. The next property would be reversibility. Most of the enzymes catalyze reactions are reversible. The reversibility of the reaction depends upon the requirements of the cell. In some cases, there are separate enzymes for forward and reverse reaction. Some enzyme catalyzed reactions are not reversible. The last property would be sensitivity to heat, temperature, and pH. Enzymes are very sensitive to heat and temperature. They are thermolabile. The maximum activity associated in nourishing protein is at traditional temperature. The correct temperature for the utmost activity is termed optimum temperature. Enzymes will be inactive at very low temperatures. This is the reason for preserving food and vegetables in the refrigerator. The enzymatic activity increases with the increase in temperature up to a certain level. At higher temperature, within the range of 60 to 70 degrees Celsius, the enzyme is destroyed or denatured, just like your boiled egg.
This time, let us proceed with our next topic, the enzyme substrate complex. Nearly all enzymes are globular proteins. Each has a unique three-dimensional shape that recognizes and binds a small group of reacting molecules, which are called The substrates. The tertiary structure of an enzyme plays an important role in how that enzyme catalyzes reaction. The following series of reversible reactions represent the steps in an enzyme catalyzed reaction. The first step, it involves the encounter of the enzyme with its substrate and the formation of an enzyme substrate complex. The part of the enzyme that binds with a substrate is called the active site. The characteristics of the active site that are crucial to enzyme function include the following. Enzyme active sites are like pockets or clefts in the surface of the enzyme. The R groups or the side chains are a unique combination of amino acid residues within the active site. These groups in the active site are involved in catalysis and are called catalytic groups. Just like pieces of a puzzle, the shape of the active site is complementary to the shape of the substrate. That is, the substrate fits neatly into the active site of the enzyme. An enzyme attracts and holds its substrate by weak non-covalent interactions. When we say non-covalent interactions, it does not involve the sharing of electrons, but rather involves more dispersed variations of electromagnetic interactions between molecules or within a molecule. Figure 11a shows the lock and key model devised by Emil Fischer in 1894. It assumes that the enzyme active site has a rigid structure that is precisely complementary in shape and charge distribution to the substrate, just like a jigsaw puzzle or a key into a lock. Or, on the other hand, the induced fit model proposed that the active site does not have a rigid shape. Instead, as the enzyme binds to the substrate, the shape of the active site conforms to the shape of the substrate, as shown in Figure 11b. This produced the perfect enzyme-substrate fit. Specificity of the enzyme-substrate complex. For an enzyme-substrate interaction to occur, the surfaces of the enzyme and the substrate must be complementary. It is this requirement for a specific fit that determines whether an enzyme will bind to a particular substrate and carry out a chemical reaction, which means that enzyme is restrictive in its choice of substrate. Let us take the enzyme urease for example. It catalyzes the hydrolysis of urea to carbon dioxide and ammonia. However, methylurea was not affected with the urease despite being structurally similar to urea. Not all enzymes exhibit the same degree of specificity. Four classes of enzyme specificity has been observed. First is the absolute specificity. These are the enzymes that will catalyze a particular reaction for only one substrate. It is the most restrictive of all specificities. The second, group specificity. These enzymes will act only on similar substrates that have a specific functional group. The third one, the linkage specificity. These enzymes will act on a particular type of chemical bond irrespective of the rest of the molecular structure. This is the most general of the enzyme specificities. Lastly, is the stereochemical specificity. This type of specificity is sensitive to the substrate's optical activity of orientation. Stereochemical molecules differ in a way in which they rotate plane polarized light or orientations of linkages. More recently, a systematic method of classifying enzymes has been established. The class of each indicates the type of reaction it catalyzes. There are several classes of enzymes which will be further elaborated in the following slides of this report. The first classification of enzyme is the isoenzyme. Isoenzyme catalyze the same reaction in different tissues in the body. For example, lactate dehydrogenase, or the LDH, consists of 5 isoenzymes. Each isoenzyme of LDH has the same function, and that is to convert lactate to pyruvate. LDH1 isoenzyme is more prevalent in heart muscles, whereas LDH5 enzyme is found in skeletal muscle and in the liver. Isoenzymes can be used to identify the damaged or diseased organ or tissues since it is a marker for a particular location. If LDH1 isoenzyme was found in the blood, therefore it indicates that heart muscles are damaged. One type of enzyme regulation involves enzymes that have more than a single binding site. These enzymes are called allosteric enzymes that have active sites that can be altered by binding of small molecules called effector molecules. As shown in Figure 3, the effector binding alters the shape of the active site of the enzyme, which can be resulted into negative allosterism or positive allosterism. Negative allosterism, it is when effector binding converts the active site to an inactive configuration, while positive allosterism, it is when effector binding converts the active site to active configuration. Allosteric enzymes are the basis for feedback inhibition of biochemical pathways. This system functions on the same principle as the thermostat on your furnace. You set the thermostat at 70 degrees Fahrenheit, the furnace turns on and produces heat until the sensor in the thermostat registers a room temperature of 70 degrees Fahrenheit. It then signals the furnace to shut off. Feedback inhibition usually regulates pathways of enzymes involved in the synthesis of a biological molecule. Such a pathway can be shown schematically as follows. In this pathway, shown in the figure, the starting material A is converted to B by the enzyme E1. Enzyme E2 immediately converts B to C and so on until the final product F has been synthesized. If F is no longer needed, it is a waste of cellular energy to continue to produce it. To avoid this waste of energy, the cell uses feedback inhibition, in which the product can shut off the entire pathway for its synthesis. This is the result of the fact that the product F acts as a negative allosteric effector on one of the early enzymes of the pathway. For instance, enzyme E1 may have an effector binding site for F. When F is present in excess, it binds to the effector binding site, causing the active site to close so that it cannot bind to substrate A. Thus, A is not converted to B. If no B is produced, then there is no substrate for enzyme E2, and the entire pathway ceases to operate. The product F has ultimately turned off all the steps involved in its own synthesis, just as the heat produced by the furnace is ultimately responsible for turning off the furnace itself. The third classification of enzyme are the multi-enzyme. According to von Dorena, he defined multi-enzyme as a protein processing more than one catalytic function, contributed by distinct parts of a polypeptide chain or by distinct subunits or both. A study by Cornish-Bowden 2004 discussed that multi-enzyme processes use two or more enzymes to catalyze reactions in a defined pathway via a cascade, a parallel, or a network configuration. Historically, the term multi-enzyme has most commonly been used to describe the metabolic activity of living microorganisms using established, modified, or de novo pathways in the context of biosynthesis. However, for organic synthesis, multi-enzyme processes outside the cell hold particular promise since individual enzyme expression and regulation may be decoupled from the metabolic network. In this way, at least two isolated enzymes can be combined in an optimal way, driving the synthesis towards a primary product. The fourth classification of enzymes are the ribozymes. These are RNA molecules that act as enzymes. There are 12 classes of ribozymes. First are the small self-cleaving RNAs, which consist of the 9 classes. Various self-cleaving ribozymes appearing in nature catalyze the sequence-specific intramolecular cleavage of RNA and can be engineered to catalyze cleavage of appropriate substrates in an intermolecular fashion, thus acting as true catalyst. The second one are the group 1 introns. These are intervening sequences that have invaded tRNA, rRNA, and protein coding genes in bacteria and their phages. The third one, the group 2 introns, are mobile ribozymes that self-splice from precursor RNAs to yield excised extranulariate RNAs, which then invade new genomic DNA sites by reverse splicing. The last one are the ribonuclease P. Ribonuclease P is a ribonucleoprotein particle that catalyzes maturation of the 5' end of transfer RNA by cleavage of precursor-specific sequences. Ribozymes have the intrinsic ability to break and form covalent bonds in RNA molecules. In many ways, they can be compared to the protein enzymes which catalyze cleavage of peptide bonds in other proteins or peptides. However, ribozymes can also be cis-acting since the ribozyme component of an RNA molecule can cleave at a specific site in another part of the same molecule. All except one of the naturally occurring ribozymes, which was shown in the table, are of the later type. We are down to the fifth classification of enzymes. Lysozymes are a class of enzymes with antimicrobial properties that are widely found across the animal kingdom as a natural bactericide. Lysozyme 1,4-beta-N-acetylmuramidase is an enzyme that plays an important role in the prevention of bacterial infections. It does this by attacking a specific component of certain bacterial cell walls, peptidoglycan. Peptidoglycan is composed of the repeating amino sugars N-acetylglucosamine and N-acetylmuramic acid, cross-linked by peptide bridges. Lysozymes acts by hydrolyzing the bond between NAG and NAM, increasing the bacteria's permeability and causing the bacteria to burst. The primary structure of lysozyme is a single polypeptide containing 129 amino acids. In physiological conditions, lysozyme is folded into a compact globular structure with a long cleft in the protein surface. This cleft is the active site involved in binding to the bacterial carbohydrate chain and subsequently cleaving it. We are now down to the sixth classification of enzymes, the abzymes. An abzyme from antibody and enzyme, also called CATMAB, from the words catalytic monoclonal antibody. They are a monoclonal antibody with catalytic activity. Abzyme binds strongly to the transition state with high association constant, enhancing their reaction state. They reduce rotational entropy and are capable of hydrolyzing proteins, DNA, RNA, polysaccharides, and many more. Abzymes has two types, protabzymes and DNA abzymes. Finally, we have here the coenzymes. Coenzymes provide additional chemically reactive functional groups besides those present in the amino acids of the apoenzymes. These are either small organic molecules or inorganic ions. Metal ions often act as additional cofactors. When we say cofactors, it is a non-protein chemical compound or metallic ion that is required for an enzyme's role as a catalyst. A metal ion cofactor can be bound directly to the enzyme or to a coenzyme. A small organic molecule acting as a cofactor is a conjugated enzyme. Coenzymes are derived from vitamins or vitamin derivatives. Example of vitamins that acts as a coenzyme are the B vitamins. Vitamins as coenzymes. Many enzymes require B vitamins as coenzymes since it allows the enzyme to function. Coenzymes serve as temporary carriers of atoms or functional groups. Coenzyme provides chemical reactivity that the apoenzyme lacks, important to metabolism reaction to release energy from food. B vitamins don't remain permanently bonded to the apoenzymes. After the catalytic action, the vitamin is released and can be repeatedly used by various enzymes. This recycling reduces the need for a large amount of B vitamins. This time, let us discuss the kinetics of enzyme-catalyzed reactions. Enzyme kinetics is the study of chemical reactions catalyzed by enzymes. Like other catalysts, enzymes provide an alternate pathway from substrate to product with a lower enzyme activity, yet the enzyme does not alter the equilibrium between substrates and products. Let us consider a single substrate reaction, which was shown in the figure. First, the enzyme must bind reversibly with its substrate to form an enzyme-substrate complex in a relatively fast reversible step. Then, the enzyme-substrate complex breaks down in a slower second step to yield the free enzyme, and the reaction product is P. Effect of enzyme-substrate concentration. For a given amount of enzyme, a reaction will go faster as the amount of substrate increases. However, when all of the enzyme molecules are combined with substrate, a maximum reaction rate is reached. At this point, the enzyme molecules are saturated and there can be no further increase in rate even if more substrate is added. Figure 19 shows a graph of initial velocity against initial substrate concentration. At contrast, total enzyme concentration for a single substrate enzyme catalyzed reaction. Take note that the curve is distorted to demonstrate the approach to the maximum velocity, which in fact is only achieved at infinite substrate concentration. The rate of an enzyme-catalyzed reaction is therefore proportional to the substrate concentration only at a low substrate concentration. As the substrate concentration is increased, the reaction rate falls off and no longer depends on the substrate concentration. Next, we have the Michaelis-Menten equation. The Michaelis-Menten kinetics is one of the simplest, best-known models of enzyme kinetics. It is named after German biochemist Leonor Michaelis and Canadian physician Maud Menten. So, in the given equation above, Km is the Michaelis-Menten constant. The equation relates to V sub 0, or the initial velocity, Vmax, or the final velocity, and substrate concentration, which is denoted by S. Enzyme concentration is incorporated in the Km. Thus, the Michaelis-Menten constant Km is equal to the substrate concentration at which the reaction rate is half of its maximal value. Refer to the figure given, which is figure 11. Km is independent of enzyme concentration. The lower the value of Km, the higher the affinity of the enzyme for the substrate. That is, the substrate will bound more tightly by the enzyme and most probably will be more efficiently converted to product. Next is the determination of the Michaelis-Menten constant and the maximum velocity Vmax. With the hyperbolic curve of the Michaelis-Menten, it is not possible to characterize the enzyme in terms of Vmax and Km, so the equation of the hyperbola is transformed into one of straight line by Lineweaver-Burke and Adey-Hofstee and Haynes. Now, we have the Lineweaver-Burke equation. It is a reciprocal form of the Michaelis-Menten equation. Take a look at the figure here. It is a double reciprocal or Lineweaver-Burke plot. The values Vmax and Km are obtained from the intercepts of the ordinate 1 over Vmax and of the abscissa -1 over Km respectively. AD-Hofstee equation is another reciprocal form of the Michaelis-Menten equation. The Michaelis-Menten equation is algebraically rearranged into the y and x intercepts corresponds to the Vmax and the Vmax over Km respectively. Lastly, the Haynes equation. The Lineweaver-Burke equation, based in turn of the Michaelis-Menten equation, is multiplied throughout by S sub 0. The AD-Hofstee and Haynes plots are favored by enzyme kinetics, but the Lineweaver-Burke equation continues to be widely used by enzymologists in general. So, all the techniques mentioned, namely the Michaelis-Menten, Lineweaver-Burke, AD-Hofstee, and Haynes, make it possible to study the kinetics of enzymatic reactions. Each enable one to characterize an enzyme experimentally by obtaining the values of the Km and Vmax for a particular enzymatic reaction. This time, let us proceed with the regulation of enzyme activity. Enzyme activity is often regulated by the cell. Often, the reason for this is to conserve energy. If the cell runs out of chemical energy, it will die. Therefore, many mechanisms exist to conserve cellular energy. For instance, it is a great waste of energy to produce an enzyme if the substrate is not available. Similarly, if the product of an enzyme-catalyzed reaction is present in excess, it is a waste of energy for the enzyme to continue producing more of the unwanted product. The simplest mechanism of enzyme regulation is to produce the enzyme only when the substrate is present. The basic concepts of enzyme regulations are: first, the cells of our body are capable of making many different enzymes. In living systems, hundreds of different enzymes catalyze reactions occur simultaneously. Regulation of enzyme activity is important to coordinate the different metabolic processes. It is also important to maintain cellular homeostasis, for example, to maintain the internal environment of the organism constant. These reactions must be regulated for the proper functioning of a living system. Regulatory enzymes exhibit increased or decreased catalytic activity in response to certain signals. An enzyme's catalytic activity can be directly controlled through the structural alterations that influence the enzyme's substrate binding affinity. Regulation of enzyme activity can be achieved by two general mechanisms. The first one is the control of enzyme quantity. Enzyme quantity is affected by altering the rate of enzyme synthesis and degradation, by induction, by repression, and by substrate metal ion and coenzyme. The second one, altering the catalytic efficiency of the enzyme, and the catalytic efficiency of enzyme is affected by allosteric regulation, feedback inhibition, proenzyme, covalent modification, and protein-to-protein interaction. Let us discuss first the control of enzyme quantity. An enzyme are protein in nature. It's synthesized from amino acid under genetic control and degraded after doing their work. Quantity of the enzyme within the cells depends on rate of synthesis and degradation. For example, the enzyme liver arginase increases after protein-rich meals. Induction of synthesis has two types: constitutive enzymes and inducible enzymes. Constitutive enzymes, it is when the synthesis don't depend on inducers, while inducible enzymes, it is when the synthesis depends on inducers. For example, induction of lactase enzyme in bacteria grown on glucose media. The third one is the repression. It is a decrease in the rate of enzyme synthesis by substances called repressors. And repression is sometimes called feedback regulation. Lastly, the concentration of substrates, coenzyme and metal ion activator. Presence of substrate, coenzyme, or metal ion activator causes changes in the enzyme conformation, decreasing its rate of degradation. Let us proceed with the control of catalytic efficiency of enzymes. Allosteric enzyme is formed of more than one protein subunit. It has two sites, namely a catalytic site for substrate binding and another site also called allosteric site, where effectors bind. Enzymatic activity can be controlled by positive and negative effectors or metabolites through non-covalent interaction. This form of control is termed as allosteric regulation. So what is positive effector or allosteric activators? This is when binding of the effectors to the enzyme increases its activity. For example, the adenosine diphosphate is allosteric activator for phosphofructokinase enzyme. The second one is a negative effector or allosteric inhibitor. It is when binding of the effectors to the enzyme causes a decrease in its activity. For example, adenosine triphosphate and citrate are allosteric inhibitors for phosphofructokinase enzyme. Glucose 6-phosphate is allosteric inhibitor for hexokinase enzyme. Mechanism of allosteric regulation. Allosteric regulation of enzymes is crucial for the control of cellular metabolism. Allosteric regulation occurs when an activator or inhibitor molecule binds at a specific regulatory site on the enzyme and includes conformational or electrostatic changes that either enhance or reduce enzyme activity. So, let us take a look at the given figures. Figure A shows the allosteric inhibition, while Figure B shows the allosteric activation. Allosteric inhibition, also known as negative allosterism, occurs when binding of one ligand decreases the affinity for substrate at other active sites. Here in the figure, an enzyme subject to allosteric inhibition is active in the uncomplex form, which has a high affinity for its substrate, which is denoted by S. Binding of an allosteric inhibitor, which is in red color, stabilizes the enzyme in its low affinity form, resulting in a little or no affinity. Figure B, the allosteric activation, is when the also known as positive allosterism occurs when the binding of one ligand enhances the attraction between substrate molecules and other binding sites. In this figure, an enzyme subject to allosteric activation is inactive in its uncomplex form, which has a low affinity for its substrate. Binding of allosteric activator, which are the green colored, stabilizes the enzyme in its high affinity form, resulting in enzyme activity. Kinetics of allosteric enzymes. When effect of substrate concentration on initial velocity or V sub zero of enzyme catalyzed reaction is studied, allosteric enzymes do not display the Michaelis-Menten kinetics. Instead of hyperbolic graph, the allosteric enzymes usually produce sigmoid saturation curve, just like what is shown in the illustration. Sigmoid kinetics usually displays cooperative interactions between multiple subunits. It also shows the property of cooperativity. For example, activity at one functional site affects the activity of the other site. Property of cooperativity comes in two ways, the positive and the negative. It is positive when the binding of one site facilitates binding of the other site. However, the negative one occurs when binding of one site inhibits binding of the other site. Metabolic pathways in a cell are regulated according to the requirement of a cell. One of the mechanisms for regulation of metabolism is determined by the amount and availability of a particular enzyme, indicating control being at the transcription or translational level. This process and regulation is associated with the enzyme regulatory molecules. Enzyme activity may be turned up or down by activator and inhibitor molecules that bind specifically to the enzyme active site. The binding site of an activator or inhibitor is reversible. It means it doesn't permanently attach to the enzyme. There are two types of inhibitors mostly affects enzyme activity. First, we have the competitive inhibition. It is when an inhibitor binds to the active site of the enzyme and blocks the binding site of the substrate. As a result, competitive inhibition increases only the Km, leaving the Vmax the same. For example, inhibition of the enzyme succinate dehydrogenase by malonate and many other pharmaceutical drugs. Second, the inhibition doesn't block the substrate binding to the active site. Instead, it attaches at another site and blocks the enzyme activity. This inhibition is said to be non-competitive inhibition. As a result, non-competitive inhibitors decreased Vmax but do not affect the Km. For example, the action of pepstatin on enzyme renin. Take a look at the given figures. The upper figure depicts the mechanism of competitive and non-competitive inhibition. Obviously, the competitive inhibition shows how other entity competes with the substrate to avoid it from binding with the active site. While in the non-competitive inhibition, the reaction is blocked since another entity was attached to the enzyme's active site, although the substrate was attached to the enzyme but not in the active site. Moreover, the lower figure depicts the graph of the competitive and non-competitive inhibition through double reciprocal plots. Our next topic is the inhibition of enzyme activity. Many chemicals can bind to enzymes and either eliminate or drastically reduce their catalytic ability. These chemicals are called enzyme inhibitors. Enzyme inhibitors are classified on the basis of whether the inhibition is reversible or irreversible, and this will be further discussed in the following slides. Irreversible inhibition. Irreversible enzyme inhibitor, such as arsenic, usually bind very tightly, sometimes even covalently to the enzyme. This generally involves binding of the inhibitor to one of the R groups of an amino acid in the active site. Inhibitor binding may block the active site binding groups so that the enzyme substrate complex cannot form. Alternatively, an inhibitor may interfere with the catalytic groups of the active site, thereby effectively eliminating catalysis. Irreversible inhibitors, which include snake venoms and nerve gases, generally inhibit many different enzymes. Reversible competitive inhibition. Reversible competitive enzyme inhibitors are often referred to as structural analogs, that is, they are molecules that resemble the structure and charge distribution of the natural substrate for a particular enzyme. Because of this resemblance, the inhibitor can occupy the enzyme active site. However, there is no reaction that will occur and enzyme activity is inhibited, as shown in the figure in the next slide. This inhibition is competitive because the inhibitor and the substrate compete for binding to the enzyme active site. Malonic acid is an example of a competitive inhibitor of the enzyme succinic dehydrogenase. Because the inhibitor has a structure that is similar to the substrate, it and the substrate succinic acid compete for the active site. As long as malonic acid blocks the active site on the enzyme, no reaction occurs. Adding more succinic acid displaces the inhibitor and reverses the inhibition. Thus, the degree of inhibition depends on their relative concentrations.
Just like any other biomolecules, enzyme can be affected by several factors. The first factor would be the temperature. Obviously, enzymes are rapidly destroyed if the temperature of the solution rises much above 370 degrees Celsius, but they remain stable at the much lower temperatures. This is why enzymes used for clinical assays are stored in refrigerators or freezers before use. In Figure 34, it shows the effects of temperature in enzyme catalyzed and uncatalyzed reactions. The rate of the uncatalyzed reaction steadily increases with the increasing temperature because more collisions occur with sufficient energy to overcome the energy barrier for the reaction. The rate of an enzyme catalyzed reaction also increases with modest increases in temperature because there are increasing numbers of collisions between the enzyme and the substrate. So, at the temperature optimum, the enzyme is functioning optimally and the rate of the reaction is maximal. Above the temperature optimum, increasing temperature begins to increase the vibrational energy of the bonds within the enzyme. Eventually, so many bonds and weak interactions are disrupted that the enzyme becomes denatured, and the reaction stops because heating enzymes and other proteins destroys their three-dimensional structure, and hence their activity. A cell cannot survive very high temperatures. What does this indicate? Thus, heat is an effective means of sterilizing medical instruments and solutions for transfusion or clinical tests. Although instruments can be sterilized by dry heat up to 1600 degrees Celsius, applied for at least 2 hours in a dry air oven, autoclaving is a quicker way. And we've done this in our tissue culture laboratory. More reliable procedure. The autoclave works on the principle of the pressure cooker. Air is pumped out of the chamber, and steam under pressure is pumped into the chamber until a pressure of 2 atmospheres is achieved. The pressure causes the temperature of the stem, which could be 1000 degrees Celsius at atmospheric pressure, to rise to 121 degrees Celsius. Within 20 minutes, all the bacteria and viruses are killed. This is the most effective means of destroying the very heat-resistant endospores that are formed by many bacteria of clinical interest. These bacteria include the genera of Bacillus and Clostridium, which are responsible for such unpleasant and deadly diseases as anthrax, gas gangrene, tetanus, and botulism of food poisoning. But why are we saying all of these? Because certain organisms known as thermophiles live in environments where temperatures range from 50 degrees Celsius to 80 degrees Celsius. Certain bacteria also live in such out-of-the-way places as coal slag heaps, which are actually burning. Others live in deep vents on the ocean floor where temperatures and pressures are extremely high. Still, others grow in the hot springs of Yellowstone National Park, where they thrive at temperatures near the boiling point of water. In order to survive in these extreme conditions, these organisms must have enzymes with tertiary structures that are not destroyed by such high temperatures. Some research shows that their enzymes are very similar to ordinary enzymes, except they contain more arginine and tyrosine. These slight changes allow the enzymes in thermophiles to form more hydrogen bonds and salt bridges that stabilize the tertiary structures at high temperatures and resist unfolding and the loss of enzymatic activity. The next factor would be the pH. Most enzymes are active only within a very narrow pH range. The cytoplasm of the cell has a pH of 7, and most enzymes function best at this pH. A plot of reaction rate versus pH for a typical enzyme is shown in figure 35. The pH at which an enzyme functions optimally is called the pH optimum. So, making the solution more basic or more acidic sharply decreases the rate of the reaction. These pH changes alter the degree of ionization of amino acid R groups in the protein, as well as the extent to which they can hydrogen bond. This causes the enzyme to lose its biologically active configuration. It becomes denatured. Less drastic changes in the R groups of an enzyme active site can also destroy the ability to form the enzyme substrate complex. Some environments within the body must function at a pH far from 7. For instance, the pH of the stomach is approximately 2 as a result of the secretion of hydrochloric acid by the cells of the stomach lining. The proteolytic digestive enzyme pepsin must effectively degrade proteins at this extreme pH. In the case of pepsin, the enzyme has evolved an amino acid sequence that can maintain a stable tertiary structure at pH 2 and is most active in the hydrolysis of peptides that have been denatured by very low pH. Thus, pepsin has a pH optimum of 2. In a similar fashion, another proteolytic enzyme trypsin functions under the conditions of higher pH found in the intestine. Both pepsin and trypsin cleave peptide bonds by virtually identical mechanisms, yet their amino acid sequences have evolved so that they are stable and active in very different environments. This is why you can eat different foods. Science does not really care much about a creator, but such amazing works help us adjust. For example, the body has used adaptation of enzymes to different environments to protect itself against one of its own destructive defense mechanisms. Within the cytoplasm of a cell are organelles called lysosomes. Christian de Dove, who discovered lysosomes in 1956, called them suicide bombs because they are membrane-bound vesicles containing about 50 different kinds of hydrolases which degrade large biological molecules into small molecules that are useful for energy harvesting reactions. For instance, some of the enzymes in the lysosomes can degrade proteins to amino acids. Others hydrolyze polysaccharides into monosaccharides. Still others degrade lipids and nucleic acids. If the hydrolytic enzymes of the lysosomes were accidentally released into the cytoplasm of the cell, the result would be the destruction of cellular macromolecules and death of the cell. Because of this danger, the cell invests a great deal of energy in maintaining the integrity of the lysosomal membranes. An additional protective mechanism relies on the fact that lysosomal enzymes function optimally at acid pH or pH 4.8. Should some of these enzymes leak out of the lysosome or should a lysosome accidentally rupture, the cytoplasmic pH of 7.0 to 7.3 renders them inactive. This is one of the evidence that our body works amazingly. The next two factors are the substrate and enzyme concentration. For substrate concentration, a given amount of enzyme, a reaction will go faster as the amount of substrate increases. However, when all of the enzyme molecules are combined with the substrate, a maximum a reaction rate is reached. At this point, the enzyme molecules are saturated, and there can be no further increase in rate even if more substrate is added. In general, the concentration of substrate in reaction is much higher than that of the enzyme. As the enzyme concentration is increased, the reaction rate increases because more substrate molecules can undergo reaction. This means that increasing substrate concentration increases the rate of reaction until the enzyme molecules are saturated. Increasing enzyme concentration increases the rate of reaction. In continuation of the factor, which is the pH affecting the enzymatic activity, we have what we call buffers. Metabolism of all biological molecules, protein synthesis, protein activation, lipids remodeling, lipid synthesis, and other biological processes all rely on the enzymatic activity. There are many factors that can affect enzymatic activity, such as including the pH. pH refers to the concentration of hydrogen ions in a system. The concentration of hydrogen ions influences the enzymatic activity by adding or removing hydrogen ions to the enzymes. As hydrogen is positively charged, it will bind to the negative charge in enzymes. So buffers are solutions that maintain the concentration of hydrogen ions in a system or a solution. There are endogenous proteins that balance the hydrogen ion concentration inside a cell and within the cell environment. These proteins are called proton pumps. However, in vitro experiments, it is necessary to have pH buffers in the system where enzymatic activity occurs because of the absence of these proteins. A mixture of an acid and its conjugated base will have the same concentration of the hydrogen ion in spite of the reaction that occurs. This only occurs when the buffer is made of a strong acid and a strong base, or a weak acid and a weak base. If either the acid or the base is stronger or weaker than its conjugate, the pH will change very slightly. The best pH buffers are the strong acid, strong base buffers or the weak acid or weak base buffers. The addition of a various compounds to a solution can decrease or increase its hydrogen ion concentration. Buffered solutions will maintain the hydrogen ion concentration regardless of the addition of the other chemical compounds. We also do have what we call colligative properties. These properties of a solution are different from those of either the pure solute or solvent. Many solution properties are dependent upon the chemical identity of the solute. Compared to pure water, a solution of hydrogen chloride is more acidic. A solution of ammonia is more basic. A solution of sodium chloride is more dense. And a solution of sucrose is more viscous. There are a few solution properties, however, that depend only upon the total concentration of solute species, regardless of their identities. These colligative properties include vapor pressure lowering, boiling point elevation, freezing point depression, and osmotic pressure. This small set of properties is of central importance to many natural phenomena and technological applications. Colligative properties are a subset of intensive properties of the solutions that depend on the concentration of solute but not the chemical identity of the solute. in accordance to Ebbing and Gammon established as 2009 and Bodner Group as 2015. For example, although the structure and mass of sugar and salt are drastically different, the impact they have on the property of solution is dependent on the number of sugar or salt molecules present rather than the sum of the molecular parts. Colligative properties influence what happens to vapor pressure, boiling and freezing points, and osmotic pressure when the concentration of solute is changed in a solution, in accordance to Okus last established 1986. So if there's a solution that requires reaction, this involves the enzymes. And the influences of colligative properties also affects the enzymatic activity. Remember, even the smallest concentration of enzymes can alter any reaction.
Finally, we've reached the end of our report. As a conclusion, enzymes have both biological and chemical attributes. Their sequences and structures delineate their role in the genome and proteome of all living organisms. Their ability to catalyze chemical reactions extends their biological function to metabolic pathways and networks. On behalf of my partner, Mark Joseph G. Pardoncillo, we would like to thank you all for listening and reaching the end of our report. Here are the literature cited for the completion of this report. If you wanted to know more about enzymes, you can use these resources. Again, that would be all, and have a great day.